Welcome to Ask the Expert, hosted by the Sugar Science, and our guest today is Ali Khademhosseini. He is the CEO of the Terasaki Institute here in Los Angeles, and it's a pleasure to have you with us.
Thank you very much, Monica. I really appreciate it.
We wondered if you could walk us through sort of your scientific background, what brought you to the Terasaki Institute, and what's going on there that pertains to type 1 diabetes. And then, we'll have sort of a little Q&A with you about, some questions we might have.
Sure, thank you. So I had a very interesting background in terms of what got me to here. So I actually studied as a chemical engineer at University of Toronto. And when I was in my third year of university, I got involved in research in a laboratory for the first time. And that was really fascinating because actually it was research very relevant to type 1 diabetes. It was about taking pancreatic islets, encapsulating them in a polymer that would basically allow this pancreatic islets not to be rejected. So it would make them stealth-like to immune cells and antibodies. I thought that was fascinating, and it really got me hooked on this whole scientific field of trying to use cells and materials and combining them. What I did after my undergraduate is I studied masters, and I studied about stem cells, particularly working on stem cell research and trying to direct the differentiation of adult stem cells. And subsequent to that, I went to MIT for my PhD, which I got in bioengineering. Now, for my PhD, I was really fascinated about the complexity of biological systems and how we can control the microenvironment of cells outside of the body using different types of engineering or material science approaches. So that kept me busy for a while, and I graduated in 2005. At which time I joined the faculty at Harvard University, and I continued doing research in the area of biomaterials and tissue engineering on a number of different disease applications, including some that are of interest to type 1 diabetes. About 3 years ago, I left Boston to come to Los Angeles. Initially, I came to UCLA, where I moved the group to this joint appointment between engineering and medical school, with the idea of trying to build bridges between the two institutions. Now, that worked out fairly well, but soon after I got to UCLA, I started interacting with the Terasaki family about the institute, with the idea of trying to really build something that is different from academia and industry in a way that it bridges the two, and with the goal of trying to enhance innovation so that we can have more of these technologies bridge the valley of death, which is kind of how you do many things in the laboratory that don't really make it to the real world. So that's what the Institute is trying to do. We have a fairly broad focus on disease topics, but we are very much interested in trying to make this translation process as efficient as possible. So some of the things that we do, which I think are fairly unique, is that we have a group of people at the Institute who work alongside investigators to vet the technology, to understand the market potential and competitors and regulatory and other barriers to entry, and try to work with investigators to perform de-risking experiments that are needed to make the different types of things that we're doing of interest to the venture community or other kind of investors, so it has a better chance of making it to the real world. So this is a little bit broadly about what the Institute does. And right now we're expanding heavily in LA area. We recently purchased our third building and trying to renovate it now to make it very leading laboratory facilities. And the kind of research that we do at the Institute, and with my background, that are of relevance to your audience, I think are in a number of different applications and in a number of different areas. One of them is trying to engineer tissues that could be used for things like transplantation or many other applications. So obviously, making tissues is something that people have talked about for many years. The whole field of tissue engineering and regenerative medicine is built on it, but it hasn't really been as, to some degree, as successful as one could have hoped for because of a couple of different challenges. One of them is because of our inability to be able to make tissues that are vascularized and can function immediately after transplantation. So we've been working on a number of different ways to induce blood vessel formations in engineered tissues, and these could be it could be pancreas, it could be heart, it could be muscle, it could be any other kind of tissue that you want to engineer. So we've been kind of taking technologies like 3D printing of cells and tissues, combining it with the latest in biology of vascularization to try to build 3D structures that are vascularized and functional. Now, we can take the same kind of principles of building tissues and apply it to creating models that could be used to make models of diseases like type 1 diabetes or other types of diseases. And the way we do that is that we basically take what we do in tissue engineering, and we can use the right cells. These would be patient cells or other types of human cells, which have much better predictiveness compared to animal models, and be able to combine those with latest in material science, biosensing, and other types of technologies to be able to make models of human disease on a chip. And why this would be relevant is that right now the cost of drug discovery is obviously is very expensive, and only a few of the drug candidates ever actually make it to the real world because most of them fail in human trials. So what we do is we actually try to bypass this inefficiency. So in the future, maybe there are better ways to come up with different types of treatments for diabetes where we can actually have these kinds of tissue models be used as predictive models to test different types of drugs or different types of therapeutics on people in a way that you tested before you actually put it on people by using these tissue models. So that's another area. And another thing that's very relevant, I think, is some of our work in biosensing and wearables. So for example, we all know that, there are things like Apple, iWatch and all these types of wearables, Fitbit. But one of the things that would be particularly interesting for particularly monitoring things like glucose levels in the body is to be able to have continuous monitoring systems that are as minimally invasive as possible. And one of the ways that we hope to do that is to develop very, very fine microneedle arrays that can be penetrating just the top layer of the skin, so it wouldn't actually reach the nerve, so they would be more or less painless. And at the same time, they can be interfaced with some of the wearable devices that we envision, so it can actually give you a real-time readout and potentially even be integrated with closed-loop feedback systems to be able to deliver insulin when needed. So, and of course, all of these things are easily can be integrated with other types of Bluetooth or other type of capability to be able to send the information directly to the clinician or to the patient, and make diabetes management and treatment as seamless as possible. So these are some of the things we work on. Of course, I'd love to chat more and have anyone who's interested visit our website at terasaki.org to learn more about some of the things that we do.
That's fascinating. Thank you so much, Ali. I had a couple of questions just sort of came right to mind. We are hoping to speak in the next few weeks with Christopher Hughes down in San Diego. He's also involved in creating an organ on a chip down there. Um, and as well, we have hosted Sumita Penether, who has been very involved with creating micro, layer needles at UCSB. So we have a bit of a powerhouse down here in SoCal. Um, I wonder, is the Terasaki Institute sort of positioning itself as a place where scientists can come to scale their ideas under its umbrella, or are you guys kind of, considering yourself as, or positioning yourself as an incubator? I mean, how does that work? If a young scientist, say, has like this great idea, hey, I have this great idea, um, you know, do they come and approach you, or do they have to be under your umbrella already working to be part of the
Yeah,
solution?
well, a little bit of both. So obviously, one of the things that I want to do is to make the institute the best place for academic entrepreneurs. So that would mean that we want to have not only the infrastructure and resources to be able to deal with people who are or ideas that are ready and people who are entrepreneurial and want to push them forward, but at the same time be able to maybe recruit people who are of that phenotype, of that interest to do the kind of work that would be translational to the real world. So it's a little bit of both. We have our own faculty currently that are expanding. We're actively recruiting additional faculty to bring in this kind of capability of being able to spin out companies or do the technology that could be licensed to existing players in the field. And the other thing is, if there are ideas, we always look at them, and if there's a really great academic entrepreneurs out there, then we would almost make positions for good people at any point.
That's really exciting. I think that we, in discussion with, with Bart Rope yesterday and Karsten Bouchard over in Denmark, we were really discussing how to, not really fast track. We don't be super fast with some of these clinical trials, but to sort of fast track some of them and scale. And that's been a problem. There's been a real, as you discussed, a sort of a gap between the bench and the marketplace. And I think, other places like the Lundquist Center in Torrance, Lindquist Center, and the ULP down in Orange County, with the Beal families behind that one, and the Terasaki Institute. I think these are really interesting places, because you're really trying to provide the bridge between the pure bench science and the marketplace, which is very important.
That's right. And as you said, there's increasing number of entities that realize the importance and the gap in the field. And realistically, at this point, the more the better, because it's such a big valley that no matter how much you try to bridge it, there is still a lot of valley left. So,
Yeah.
so yeah. So I think it definitely makes a lot of sense.
How do you have outreach programs to USC, UCLA, UCSB? I mean, is that your intent, to sort of reach into those departments and have a collaboration, or how does that work with the
Yeah,
academic institutions?
definitely. So we have a lot of existing collaborations with obviously UCLA and USC, and many other academic institutions throughout the country. So there's a long list of them. But in addition, we have a lot of interactions with industry. So we have a lot of ways in which we try to build bridges with the industrial partners, whether it's through sponsored research agreements or joint partnerships in trying to develop specific diagnostic or therapeutic approaches. So I think that we definitely want to be able to leverage the best of collaborators in both areas, whether they're industry or academia.
And so when, if we were just sort of to put out a, a call for talent, right here, would you say that there's room for talent in the two realms you talked about, the organ and the chip, and the sort of the micro layer needles that pertain to type 1 diabetes research?
Yes, so we definitely have open positions in both of those areas, as well as many other areas. So I think with the whole wearables, there's a lot of interesting things happening in both building better types of materials that could interface with the body and have less of issues with fouling and other things that some of the standard things have had, as well as the electronics aspects of being able to do all the reading and basically dealing with the data. And on the other side, with the organs on a chip, we have the ability now to work with a bunch of different techniques to take cells, including pancreatic cells, and be able to really understand their behavior, engineer their microenvironment, and then be able to use them in different applications. So I think with both of those, I think there's a lot of opportunities at the Institute, and we're always eagerly waiting to see new people who are excited about the field.
That's fantastic. I think it's a great opportunity here at the Terasaki Institute for those who are interested. Please reach out directly on our collab, where you'll find Ali. You'll be soon there as a member. And I wondered, if anyone else has any further questions for him.
Yeah, I actually had a question. Well, I think a lot of people, my background is in biomedical engineering, so a lot of people like myself know how prolific you are individually as a scientific leader. And I was just curious about maybe if you could share with us some of your secrets, or how you feel that you've gotten to such a high place in the scientific community in terms of being able to make such impact in so many different areas of research.
Sure. No, that's a great question. Obviously, like anything else, what matters at the end is who you work with. So I work with a lot of great people throughout the years, and that at the end is the best way to increase one's productivity and thought process. There's also in research, there's a very clear way of trying to do research in a way that's more efficient, where you really understand your questions and their importance, and also what is the best path to get those things answered. So so actually at our website, I give a series of fireside chats where I lay out some of these secrets. So I definitely encourage people to go through and have a look at them, because I have many hour-long sessions about this specific question.
Okay, awesome. Another question I had was in terms of the resources that are available at the Institute. I guess I'm still a little bit unclear in terms of, since it is kind of this hybrid model that you say that there's like academics, but it's also kind of incubator-ish and working with industry. So um, do people that work at the Institute and researchers that you have in your group, do they have access to like business leaders and such that they can communicate with, or like clinical leaders to figure out how the best approach is to bring technologies to market? Could you expand upon that?
Yeah, excellent question. So we have partners in both. We actually have a number of clinicians who are in our kind of affiliate faculty. So they still have their clinical work at different hospitals, but they help in guiding us to some of the clinical problems, as well as be able to have a lot of interactions with entrepreneurs and other types of people who've actually gone through the process of building products and companies, who kind of work with our investigators to give them some of their advice.
Okay, great. And another thing I was thinking about, and I'm kind of, I guess, getting a survey in terms of what people think. A lot of my background is in pancreatic islet transplantation. And so there's a lot of people in the field that think that pancreatic islets are special and they're maybe a little bit more challenging to work with than other cell types, specifically because they're organoids and they're not like single cells and they come in all different shapes and sizes and that kind of thing. But I've also heard from other groups, people that they say, you know, cells are fundamentally the same. You apply the same engineering principles and you'll figure it out. So I guess I'm curious if you're in one of those camps or maybe have a different view on it.
Yeah, so I've learned a long time ago is better not to put myself in a camp against other camps. So, so I would say there is validation in both of those statements. There's definitely unique things about every tissue, including the islets, and specific challenges about maintaining them outside of the body and trying to re-engineer them. And they do have some aspects that are very similar to other cells. The fundamental machinery and architect, like what it takes, the genomics aspect is fundamentally similar. So there's merits to both of those camps.
Okay, thanks.
Fantastic.
I would agree. Yeah. So I think there does need to be special attention to the cells and the particular challenges that you're working with, but you know, you can apply similar principles, but I think I lean more towards that they're special, but that's probably just a bias that I have. Thanks for sharing.
Thank you.
Thank you so much for, entertaining our questions. I really appreciate you stopping by today, Ellie, and we're really keeping an eye on what's coming next for the Terasaki Institute. It's very exciting times.
Thank you very much. And I look forward to hopefully hosting you guys at some point very soon when things are settled down.
Sounds great. Bye-bye.
Thank you. Thank you, everybody. Bye-bye.